Hey everybody, it's Mr. Cho, and I'll be going through the paper 4 biology for 2022. Alright, let's get started. Okay, so figure 1 shows the diagram of the eye. Okay, A, B, C, D. Draw an X on figure 1.1 to identify location of the lens. So where's the lens will be over here. This is your lens. X. Okay, so 1 done. Okay, describe how the parts labeled B and C in figure 1.1 respond when a person moves from dim light to bright light. Okay, so bright, it means that your pupil is very small. Okay, so now there's 2 marks. Okay, so I need at least 2 points. Alright, so when the person moves from dim light, is iris that is changing. Alright, so okay, so the radial muscles in the iris. Okay, what happens to the radial muscles in the iris? So here is my eye. Like so many of these things. This thing. Okay, so this is my pupil, right? And then if it's contracting, so it's very small pupil, right? Pupil inside here. Okay, so the radial muscles must extend out. That means they must relax. These are radial muscles. And then the circular muscles, right, they all have to contract, right, to pull it towards the middle. Okay, so the radial muscles in the iris will relax, and the circular muscles will contract. The pupil, okay, so I forgot to label. So iris is B, right? The pupil, C, will become smaller in size, and this will reduce the amount of light that is entering into the entering through the lens, the retina. Okay, so that is 1 mark for this, and then 1 mark for the pupil. Okay, 2 marks. Okay, so describe how the parts labeled A and D in figure 1.1 respond when a person changes focus from a distant to a near object. Okay, so near object is less than 6 meters. Okay, so like something like a watch or something. Okay, so what is changing? A and D. A is our suspensory ligament. D is the ciliary body, which contains ciliary muscles. Okay, so the okay, so near object, right? Light rays from a near object are, okay, so object is very near. So imagine I draw object here. Check. So they're very near. So these rays are diverging. Right? So they're diverging. So you need our lens to be rounder. We need our lens to be rounder so that it will refract more light. Okay, so that means that this, which is our ciliary body and it contains a ciliary muscle, it must actually contract forward. Right? Contract forward. So that our suspensory ligaments will become very slack. Okay, so they slacken, and then this whole lens will become rounder. Okay, so light rays on diverge are divergent. The ciliary muscles in the ciliary body, which is alphabet D, will contract and move nearer to the, that's right. Okay, so that will cause the suspensory ligaments to slacken. Okay, so what's the suspensory ligament letter again? It is A. Okay, the suspensory ligaments to slacken. The lens will become more convex. Okay, so convex means how round it is. So if it's more rounder, then it's more convex, and thicker to focus or to refract more lights onto the fovea, the retina. Okay, so 1 mark for my ciliary bodies, right? And then 1 mark for my suspensory ligaments. Probably another, maybe a minus mark if I don't write about my lens becoming more convex and thicker. So this is done. Okay, state the part where the focus image is formed. Okay, where is it? The fovea of the retina. So question 2, figure 2.2 shows 2 different food chains. Figure chain 1 is grass, rabbit, fox. Okay, so here we have 3 trophic levels. Food chain 2, oak tree, snail and thrush bird, and hawk bird. There are 4 trophic levels. Okay, so name the producers. Producers are those that are able to produce their own food. So it's the grass and the oak tree, the plants. So a herbivore will be either the rabbit, snail. Okay, outline 3 ways in which energy is lost between the trophic levels. Okay, so energy is lost. Energy is lost as heat during the process of respiration. Energy is lost. Oh yeah, my spelling. Oh my gosh. Okay, my energy is lost in the unconsumed parts of the prey. Energy is lost in the excreted waste, such as feces and urine. Okay, so 3 marks. Here. Okay. Explain why food chain 1 can support a greater number of top carnivores than food chain 2. Okay, so food chain 1 has only 3 trophic levels, whereas as compared to food chain 2, which has 4. 90% of energy is lost between each trophic level. So there is less energy available for the top carnivore in food chain 2 as compared to food chain 1. Okay, double check that I'm talking about the right thing. Okay, that's correct. 2 marks. Okay, an insecticide was sprayed on oak tree in figure 2.1. Eventually, the insecticide caused the hawk bird population to decrease. So this is known as bioaccumulation. Okay, so explain why the insecticide affected the hawk population but not the snail. Okay, so it's not that the hawk eats the snail directly. The snail is eaten by other consumers, and then subsequently the hawk eats those consumers. But this is known as bioaccumulation. Okay, so non-biodegradable insecticides will pollute the soil and water. Plants will absorb it, the pest insecticide, into their bodies, which will then be consumed by the snails. The concentration of insecticide is not enough to harm the snails. However, the hawks will consume. Okay, so however, the concentration of insecticide increases up the food chain. Over time, the hawks will accumulate large, high concentrations of insecticide in their body, which can lead to mutations and harmful effects. This is known as bioaccumulation. Alright, so quite long for 2 marks, but yeah, you have to do it. Okay, question 3, figure 3.1 is a diagram of human alimentary canal, associated organs, identify the liver and use a label line and a name. Okay, so label line, okay, liver. Okay, so okay, now state where the alimentary canal where ejection occur. Ejection means you poop. Okay, so this is in the anus. Okay, then where do you ingest food? Ingestion means you eat, so that's in the mouth. Where do you absorb digested food? In your small intestine. What is the question? Intestine. Oh gosh, my spelling. Okay, so question table 3.1 shows some digestive enzymes and some of their substrate and products. Complete table 3. Okay, amylase that's found in the mouth as well as the pancreatic juice, so it works on starch. The end product will be maltose. Okay, as we know, right, starch is a long chain of individual glucose molecules, long chain, but the amylase works on pairs. That means that you cut this into pairs. So a pair of glucose is known as maltose, and then of course by itself is glucose. What enzyme works on protein will be our proteases. The end products could either be polypeptides in the stomach or amino acids. Okay, let's just make sure that I'm not misleading. Yeah, that's correct. Okay, then what is the end product of fatty acid and glycerol? This will be lipids. Don't write fats. No fats. Because lipids is the right term. Okay, then substrate. What's the what am I talking about? My gosh, it's over here. The lipids is the substrate and the enzyme is the lipase. Dum. Okay, then maltase work on maltose to give you glucose. Okay, so always check, right? Starch, amylase breaks down starch and maltose. Proteases break down proteins into polypeptide amino acid. Lipase breaks down lipids into fatty acid and glycerol. Maltase breaks down maltose into glucose. Yep, that's right. Okay, now name the blood vessel that transports blood rich in nutrients from the small intestine to the liver. That will be a hepatic portal vein. Hepatic portal vein. Okay, here we have an effect of wind speed. Okay, water loss, so this transpiration. Okay, then we got the fan, we got the ruler, plant, reservoir, air bubble. Okay, so what's going to happen when the water vapor exits from the stomata? Then the air bubble will move forward. Okay, so the student measures the distance of the air bubble moving 1 minute, and the fan was placed from the chute. Distance moved by the air bubble is equivalent to water loss. Then table 4 shows the results. Okay, or this. Okay, so this one I'm not going to do. You have to do it yourself, but keywords, right? S, L, A, P. S stands for size. The size must be more than 70% of the graph. Okay, it must be more. That means that your whole drawing is not so small, right? This is a bit too small. It's not 70, it's only 50%. So your whole drawing needs to be like 70%. So it's like, so all the way, something like that. Okay, I don't even know if it's the right shape. Probably not the right shape. Okay, then we are supposed to draw a line of best fit actually, so it's probably not, may not be a curve, right? Depending on your drawing. Okay, so line of best fit. What does line of best fit mean? It means that, let's say you have some axes like that. Okay, so for line of best fit, right, you're supposed you may or may not have to touch up all the points. Okay, but you'll never do it like a roller coaster, like wow, from here connect the dots. It's not connect the dots. Okay, so line of best fit means that you're trying to balance the number of points on the left and right of the line. So it's just if it's a straight line of best fit, then it'll be straight line somewhere like that. Okay, so then you see that here I have 2 on the left and 3 on the right. Of course, there's only 5 points, so you have to 2 and 3. And so it's also like in the middle of everything, like an average of everything. Alright, there can also be line of best fit for curves. So let's say that it's like that. Okay, so you see here there's abnormality, right? So ignore abnormality, just draw the curve. Okay. Wait, wrong. The thing should not, the tail should not go out. The tail should only start from the first and then end on the last. Okay, so that's the line of best fit. Okay, of course, what else do we need? Make sure our scale, right, all the scales are split out nicely. Okay, and they're all labeled properly. And there's a label. So a label should be somewhere around here and somewhere around here. Okay, so what are the labels? Where do you find the labels? These are the labels. Okay, these are the labels. Distance of fan from plant shoot. This is what it calls your independent variable. Distance moved by air bubble. This is what you measure. So this is your dependent variable. Okay, so your independent variable will always go to the x-axis. Dependent variable always go to the y-axis. Okay, so I start by writing distance of fan from plant shoot, and then I start to split it off at the x-axis. Okay, the rest you do yourself. So S stands for size, L stands for best fit line that is not extrapolated and ignores abnormalities and is smooth. Okay, that means if it's a straight line, it's not like mine. This very ugly straight line. It's actually a ruler straight line. If it's a curve, it's a nice smooth curve. Then for the plots, it means that all these axes must be plotted on the correct position. And oh yeah, the scale, sorry, not scale, the size, line, axis. The axis, right, must also have all of these, the axis titles lah. In a sense, the x-axis title and the y-axis title. If you want to give me the overall title, also can, but honestly, I don't think that will be important. Okay, but if you got time, then just go and write it down. Okay, let's move on. So, wow, this one a bit hard if I never draw the graph. Describe and explain the effect on the results in table was repeated at greater humidity. Okay, so greater humidity means that the rate of transpiration will drop. Okay, so describe and explain the effects on the results on in table 4.1. Okay, so the higher the humidity, the lesser the rate of transpiration. This is because the concentration gradients between the intracellular air spaces where the water evaporates and the atmospheric air is not as steep. Okay, or you can also just write it's gentle, it's too gentle. Okay, thus and describe and explain the effects on the results. So the results in table 4.1 will see a overall increase. See an overall increase, not overall. An overall increase. Then how about the distance moved by the air bubble? this an increase. Okay, no, not increase, my bad. We'll see an overall decrease in the movement of the air. Okay, so how do I write? Air bubble. For example, the distance. Okay, I don't think I need an example. I think that's already enough. Okay, so they are described and they are explained. Okay, so I described that it will be decrease in the movement of the air in the movement, and then I explained because the higher the humidity, the lesser the rate of transpiration. Okay, so that should be 2 marks already. Okay, state the part where the water vapor would exit the leaf. Okay, give me a moment, answer some. Okay, so where was I? I was saying that the water vapor exits the leaf from the stomata. Okay, not the gut cells. The gut cells are surrounding the stomata, but the stomata, that's the hole where the water vapor would exit the leaf. Okay, how does water move through the plant stem? Okay, there's 2 processes: capillary action and transpiration. Okay, so how does it move? Okay, so water is initially brought up the plant stem by capillary action. Will be so then I say as water evaporates from the stoma. So the stomata and exits the leaf. This creates a force known as transpiration pull. Transpiration pull will cause the water in the plant stem to move up towards the leaves. Okay, so that's my 2 marks. Okay, question 5. Photograph of a person with dimples. So how many people cast at dimples? 16 people has dimples. 8 people has no dimples. Calculate the total number of students. What kind of stupid question is this? Just add it together lor. Right, 16 plus 8, 24. Okay. Is it too easy? Quite right. 16 people has dimples. 8 people don't have dimples. Calculate the total is 24. Okay. So describe the evidence from figure 5.2 that the presence of dimple is an example of discontinuation. Okay, so what does it mean discontinuous? Discontinuous variation, I spell only. Sorry, you all don't have autocorrect in the exam. Discontinuous variation is when there are only a few groups or categories for a certain traits. Okay, as seen in figure 5.2, there are only 2 groups, either with dimples or without dimples. Okay, a couple wants to calculate the likelihood of their offspring inheriting dimples. It's thought that the allele for dimple is dominant. Okay, this is not scientifically proven yet, it's just thought only, but it's big D. Then allele for no dimple is recessive, and then raised by small d. Okay, sure. Both parents have a heterozygous genotype, which means that it's one big, one small, and then complete. Okay, so both parents have one big D, one small d, one big D, one small d. So then you got big D, big D, big D, small. Okay, let's put the big one first. Don't put the big one behind the small one. So big D, then small d, small d. Okay, easy 2 marks. Okay, now just double check. Correct, right? Heterozygous genotype, heterozygous. Okay. Use your answer to calculate a percentage chance of the offspring inheriting dimples. Okay, so chance means percentage. So I see that there's only one child here, or one potential child, so it's 25%. Okay, what are the conditions caused? Some conditions are caused by mutation in the structure of a gene. Okay, so define the term gene. A gene is a sequence of nucleotides that is found on a chromosome. It codes for a single polypeptide. Okay, then one disease caused by a mutation in the structure of a gene. Okay, so there are two types of mutations. One is the number, which is Down syndrome. Then the structure will be sickle cell anemia. Okay, then we have a student investigated the effect of pH on enzyme activity at 30 degrees. Then figure 6 shows the summary results. So describe the results shown in 6.1. Okay, so for pH, right, pH, we already start the extremes, right, which is around here. These are the extremes 2 and about 10, and then we go up the middle, and then we go to the optimum in the middle. Okay, so describe the results as shown in figure 6.1. At extreme pH of 2 and 10, the enzyme is completely denatured. There is little to no enzyme activity. Okay, why I put little is because they never give me numbers here, so it'll be hard to see. I'll expect it actually to be falling down all the way to the bottom, but it's okay, never mind. Okay, so as the pH moves towards the optimum of 5.5, the enzyme activity starts to increase. Then at the optimum pH of 5.5, okay, 5.5, yes, okay, optimum pH of 5.5, the enzyme activity is the highest, which means the rate of reaction is the highest. Okay, so 2 marks there. Okay, now we have the investigation is repeated at 80 degrees Celsius. Okay, so it says there's no enzyme activity, explain why. Okay, at 80 degree, okay, I can't draw that small circle, so 80 degree Celsius, remember to draw that small circle. The enzyme is completely denatured. Okay, so why now we're going to use the lock and key hypothesis to explain why. So when the enzyme is denatured, there's no more active site, right? When the enzyme is denatured, the active site is lost. The substrate cannot fit into the active site, like how a key will fit into the lock. There are no enzyme substrate complex being formed. I spell it separately. Thus, there will be no enzyme activity. The substrate will not be broken down into its products. Hold on, let me answer the question. Okay, now let's do section B. so here section B only need to answer 2 questions. I will start by quickly scanning through. Okay, so I see that question 7 is about plant cell, animal cell, so it's just structures. Okay, so that should be quite easy, just our 3 main structures and the function. Then we have osmosis question, because that's pure water. Then we talk about what happens to the cells in the different solutions, so it's osmosis question. Then should be quite easy. I will do section 7. Okay, then question 8 is about long run breathing rate increases, gets a pain inside, muscle cramp. Okay, so this is about lactic acid, and then also about anaerobic respiration, basically. Okay, so this one is easy, but I think I will not do this. Okay, then of course if I'm not doing that, I'm not doing this also lah. But just to say, this is about pathway of carbon dioxide molecule from the blood in a capillary to exhale. Okay, this one is quite doable. Then question 9, it says blood concentration of estrogen and progesterone. So this is about reproduction in the menstrual cycle. So describe and explain the changes in the ovaries and the lining uterus using information about hormones. Okay, this one quite easy. Then after that, hormonal coordination. Okay, it's also very easy. Okay, so let's do 7 and 9 first. Okay, I'll do 8 later. Okay, so let's start by typing. Okay, so a typical animal cell and a typical plant cell both have, okay, because I'm talking about what's common, right? What's common? Both have a nucleus, ribosomes, cell wall, cell wall blah, not cell wall, cell membrane, cytoplasm, okay, DNA, and what else do they have? Ribosomes, cell membrane, cytoplasm, DNA, it's found in the nucleus. They don't. Plant cells have cell wall, which animals don't. Then nucleus already. Anything am I missing? Cytoplasm. Okay, that should be more or less it. Okay, so function of the nucleus. The nucleus contains genetic material like DNA, which controls the cell growth, repair, and division, and cell division. Okay, then we have the ribosomes. Synthesize. I forgot one thing. Yeah, no wonder I keep thinking. Mitochondria. Okay, so the ribosome synthesis proteins from the basic unit of amino acids. The cell membrane controls the movement of substances in and out of the cell. The cytoplasm is the site of most chemical cell activities and chemical reactions. Okay, then the mitochondria. The mitochondria is the site of aerobic respiration. Okay, and that's 4 marks. Okay, then we have our cells are immersed in pure water. So describe and explain the similarities and differences. Okay, so pure water has a higher water potential as compared to both animal and plant cells. Okay, so there will be a net movement of water molecules from the pure water into the cells by osmosis through a partially permeable membrane. This will cause the plant cell to become turgid, right? And swollen. Okay, just turgid. Because as it has a cell wall which will prevent it from bursting. However, the animal cell will lyse as it does not have a cell wall. Okay, so similarities, both animal and plants will increase in size. Differences: plants cell will become turgid, but animal cell will become lysed, or you can also just say burst. Okay, is there any other differences? Describe and explain the similarities and differences that will occur to the animal cell and plant cell members in pure water. Okay, so blah blah blah. I got my osmosis statements. I got my different similarities. Is there any other similarity? Similarities. Oh, okay. Water moves into the cell through their cell membrane. Okay, that's one more similarity. Okay, so I think that should be the 6 marks already. Okay, now let's talk about question 9. Question 9, we have blood concentration of estrogen and progesterone. So estrogen is at the start, progesterone is later. Then describe and explain the changes in the ovaries and the lining of the uterus using the information. Okay, so these are must use information. Okay, so from days 1 to 5, the lining, the uterine lining, blood, and the unfertilized egg are shed during menstruation. Estrogen is released and it helps to repair the uterine lining from days 6 to 10. Day 11 to 17 is the fertile period. Estrogen level increases sharply, and the peak of estrogen at day 13 will result in the ovulation on day 14. Okay, subsequently, the estrogen level will decrease. From days 18 to 28, the progesterone level increases in order to further thicken and grow the uterine lining, as well as to prevent a second ovulation. Okay, so 1, 2, 3, 4, 5. Okay. Okay, next question. Okay, describe how the hormonal coordination differs from nervous coordination. Okay, so there are 5 points. Hormones are hormonal coordination in IO spelling involves hormones which are carried through the blood, whereas nervous coordination my spelling is so bad, involves nervous impulses that move through the neurons. Okay, then hormonal is usually slow, usually fast. Okay, it's very fast. Yes. Hormonal coordination can last for a long period of time. Okay, let me pause a while. Okay, then of course, oh wait, I realized I missed something here. Hormonal coordination is usually a slow response. So okay, then lastly, hormones can hormonal coordination targets a few organs, usually targets a few usually targets a few organs, whereas nervous coordination targets just one organ. Okay, usually. Okay, 1, 2, 3, 4, 5. Okay, because blood and neurons also is 1. Okay, so that's 5. Okay, so let's run back to the question that we skipped. Okay, so a student goes on a long run, period increases, gets pain in the side. This is muscle cramp. Okay, so during a long run, the energy required exceeds what only aerobic respiration can provide. Anaerobic respiration will occur to provide more energy. Okay, the product of anaerobic respiration is lactic acid and a small amount of energy from glucose. Okay, so lactic acid has been proposed to cause muscle fatigue, muscle cramps, soreness, and pains. Okay, I feel like I missed out something important here. 6 marks. The breathing rate. Okay, maybe I missed out the part about breathing rate. I need to restructure my thing a little bit. Okay, so what should I say? Okay, so during a long run, the breathing rate increases to provide more oxygen to the lungs for aerobic respiration. However, there is a maximum breathing and heart rate. When the energy required exceeds what only aerobic respiration can provide, then aerobic respiration will occur to provide more energy. The product of aerobic respiration is lactic acid and a small amount of energy in the substrate glucose. Lactic acid is proposed to cause muscle cramps, soreness, and pains. Okay, so after some time, the lactic acid will be transported from the muscles to the liver, where it can be converted back into glucose or excreted excreted through the kidneys. Okay, so now we're going to describe the path of the carbon dioxide molecule from the capillary to the alveolus. Okay, so there is a high concentration of carbon dioxide in the blood capillary and a low concentration in the, pardon my spelling, alveolus. This creates a concentration gradient, and the carbon dioxide will diffuse out from the capillary into the alveolus through the alveolar wall, which is one cell thick. Okay, then the carbon dioxide molecule will be pushed out during exhalation as the pressure in the lungs is high. It will move from the alveolus through the bronchioles, then through the bronchus, up the trachea, then from trachea, and finally out of the nostrils or the mouth. Okay, did I miss out anything here? Should be okay. Okay, that should be the 4 marks. Okay, and with that, we have finished. Okay, what I will do next is I will run through all what I've written. Okay, so this paper is 1 hour, like 15 minutes, right? Which means that I only spend 15 minutes here. I have about 20 minutes to check. So I will check everything, make sure that I count the number of marks that I more or less hit every single question. I check all the way to the end, make sure I didn't miss out anything, check all the way to the top. If I, for some reason, if I did all the question, I need to cancel this. So if I do this, right, I must cancel 8, cancel, because I'm not submitting this part. In fact, if you want, just cancel the whole page. Cancel the whole page. Okay, so then just check everything. If you have really have enough time, personally, what I would just suggest for you to do, take like a 1 minute mental break, close your eyes, everything, then open it again and run through everything as if you've never done it before, right? So you'll avoid careless mistakes. Make sure that you you check all the marks, right? For example, this one, you must draw the X, so you put a dot on every single mark that you do to make sure you do everything. Okay? Alright, so with that, all the best, and I'll see you in school. Bye.